everyone, and hello Karen. Thank you so much for joining me today as we go down the rabbit hole and take a look at some of the niche and unusual aspects of genomics. Today we're going to be talking about crop science with the lovely Karen, so if you could just introduce yourself and tell us what you do.
Sure, thanks for having me. My name is Dr. Karen Massel, and I'm a Canadian who did my PhD out here in beautiful Australia. So I traded the snow for the sun. And I started my PhD career developing genome editing systems in sorghum bicolor, which is a relatively unknown crop, but it's a really important one for food security because it feeds about 500 million people. And I'm now working as a postdoctoral research fellow on this ARC discovery project, which is about looking at drought tolerance in both sorghum and barley, which has been really exciting.
So obviously today we're going to be talking about crop science, but I think maybe it'd be good to kind of just start off from the basics and what is crop science?
Yeah, exactly. Crop science, it's a huge field. I think it's very interdisciplinary. It's sort of at the end of the day, it's all about studying plants that have really important sort of economic uses and commercial uses. But this has to bring together so many different aspects of science. So in crop science, we often talk about our G by E by M, which is genotype by environment by management. And all three of these aspects need to be researched in order for us to really improve on crop production and understanding how crops function. So I'm a lot of the times working on the G aspect, working on improving the genetics behind it and how that relates to the environment and management styles. But you also need to bring in people who work on soil science, understanding what's going on in the soil and all the nutrients. It involves plant breeders. You need people working on different types of weeds. You need pathologists working on all the different viruses and insects that are attacking your crops. And nowadays with sort of big data, we've got lots of bioinformatics, we need computer scientists, we need people modeling for different environmental conditions and different genotypes. So it's a really big field of science to be a part of, which is great.
Yeah, like a really big kind of team effort.
Yes, definitely.
Yeah. What are some of the kind of like, obviously there's probably loads, but what are some of the kind of main aspects of crop science, like where it can be like applied?
Yeah, so the application of crop science, I mean, I guess at the end of the day, we're trying to produce more, or at least produce something different. So people probably don't realize exactly where their food is coming from, for example, but it's all about sort of generating different varieties that are being grown. So when you go to the grocery store, it's not just an apple, you've got all your different types of apples that you can now eat, and that's all from plant breeding. So that's one sort of aspect. Crops also have really big uses in the industry sort of fields as well. So we can make biofuels to try and get away from sort of oh my God, I can't think of the word. Oh God.
Fossil fuels.
Yeah, exactly. All of the natural resources. And even, we use potato starches to, stick in your collars of your stuff. So I think crops have sort of really big uses. And also I think understanding just how plants function sort of in the natural world is also really important. So that sort of core biology of how do things function is also really important. That's less applied, I guess. But.
Yeah. So you said that you're kind of working on kind of the G aspect. What kind of work are your team currently looking at?
Yeah, so my work is so I've got my fingers in a lot of pies. So I do have quite a few different things that I'm working on. My main project right now is to work on CRISPR Cas9 genome editing, which we're trying to manipulate a lot of these genes involved in how the plant grows. So we're trying to reduce sort of how the carbon is organized in the plant, trying to reduce biomass, which will then lead to more grain yields, especially under water stress, which is really important if you want to produce more in some of these lands that are really difficult to grow plants on. I also work on a lot of for traits that are involved more in reproduction. So if we can try and make the plants have more flowers, which in turn lead to more grain, we can try and boost yields that way. I also work on some really interesting projects where we are looking at the different types of pollen that we're finding throughout Australia. So we can try and have sort of like an allergy database that you can sense when the things that sort of cause your allergies, when they're sort of spiking. Because in Australia, where it's sunny all the time, we get allergies 365 days a year, and it's all from different plant species. So I'm part of this project where we're studying the genetics of the pollen that we find in the air, and hopefully we can have sort of an app for so you know when you need to take your allergy medicine.
Yeah, I don't know what's going on over here because the weather's so bad, but my hay fever is always so bad. So, how kind of the obviously you said you're working on CRISPR. How are these kind of advanced technologies helping you to kind of be more precise in this area?
CRISPR-Cas9 has really opened the doors for how we can do genetic engineering. I think it's one thing to be able to put a foreign gene into another plant, but using this sort of technology, you can create really precise changes exactly where you want them to be. And even if there is, I mean, we're not finding that we're getting that much off targeting, but I know when you talk to people in medical sciences, they're very freaked out about all the off targets. But if you can imagine in a plant, no matter what sort of crop species you're working on, you're probably going to end up breeding with it anyway, which means you're removing a lot of that material outside of the region of the genome that you're manipulating. So you don't even really care if there's off-target mutations because that part of the genome is not going to be in your final product anyway. So I think working on plants, there's a lot more potential for this sort of technology just by the nature of the work itself.
Yeah. What kind of other kind of genomics tools are you guys kind of using to help within the field?
Oh, I mean, there's so many genomic tools. I mean, so obviously, so sequencing can be difficult. Well, maybe you have to take a step back even further. So plant genetics are complicated. I think people underestimate how complex they can be. A lot of plant species haven't even been fully genome sequenced just based on the complexity. So the human genome's like 3.2 gigabase pairs, I think is what everyone says. So wheat, for example, I think is 14 and a half. And it's a hexaploid, which means that you've got in that sort of jumble, every gene, there's 3 different allelic copies, and some of them can be so similar that it's hard to know, are they from the A, B, or D genome? And then you've got crops like sugarcane, which are an alloploid, which means we don't even know what ploid level they are at. And a lot of them will be 99% similar to each other, these genes. So it's really hard to even get some really great genetic data on some of these crop species because the ploidy level makes it complicated and their shear size makes it complicated. But it also means that but because we can breed with crops and we can actually create populations and we have, you can sort of go back to the domesticated lines, all these wild cultivars, and you can compare wild to domesticated and improved lines. We can get a lot of data about how sort of the evolution of these crops have come through. You can use things like genomic selection for breeding practices, and we use modeling. It's I think in the field of genetics, crop science is definitely a really interesting field where you can do a lot. Mostly just because you can grow 300 plants, cross them with whatever parent you want. No one cares, when you're working on plants. So you have a lot more potential to really do sort of QTL analysis and actually pinpoint the genetic basis of specific phenotypes.
Yeah. I know you said it's difficult, but are there any sort of like, I don't know this area that well, but are there any sort of attempts to kind of sequence the genomes of like different plants and then create like a catalog? Because like you have like, for example, like the vertebrate catalog where they're trying to sequence the genomes of different vertebrates, and then they're going to put it in like one big catalog. Is there attempts to do that within the plant field? I don't know too much.
We definitely know we can sort of follow a lot of like the synteny between different families. So you can go to the sorghum genome, and there's a lot of online programs you can find. It's sort of homologous gene in maize, and you can sort of see how the chromosomes have been rearranged over throughout evolution. And there's a lot of work now. I mean, I read a paper recently published by some colleagues of mine where they do pan genomes, which means that you have your sort of your reference genome, which is fantastic if you can have it. But then when you go to sequence a different cultivar, and if it's not very related, it means that there's all these regions that you can't actually match to the reference genome. So the pan genome then takes those parts into consideration. So it's sort of like, because one of the issues with crop breeding has been that throughout domestication, we've really limited the diversity of the genetic pools, which is why we're always trying to reintroduce diversity, because diversity is sort of how we are able to adapt to specific, the environments and different pathogens. And so a lot of times we want to know those sort of regions that have been lost in the domesticated lines from wild cultivars. So doing pan-genome sequencing is one of the new things to be doing, especially for some really important crops.
What are some of the kind of challenges working with crops? Because obviously you said that you can kind of make plants with any other kind of, you can breed them together. What are some of
Yeah.
the challenges with working with plants?
I would say, so I guess it depends maybe on what your research is specifically within the field. I know for myself, because there's sort of only a few main crops that people really invest a lot of money in. It means that there's a lot of the research is sort of focused on, we've got maize or corn, as people call it if you're American. You've got wheat and maybe barley people have heard about, but a lot of these sort of orphan crops is what we call them, which do feed. I mean, everybody likes to go to the grocery store and see all of those different vegetables and all the different flours. Everyone wants the gluten-free alternatives, but no one really, I'm sure most people couldn't really think of what's a gluten-free cereal, like what is that flour that they're sort of eating. So I'd say it's hard sometimes to get funding for sort of, if you're not in the top 5 important crop species, to do the work, and there's just so much less known about a lot of these sort of features.
Yeah.
But being able to, crops are also complicated. I mean, you have to grow them. They all like their own temperature, their own humidity, their own nutrient profiles. Some of them are a bit more of a diva than the other ones. There's a few things to work out every time you switch a crop.
So you kind of look at kind of the genomic side, but are people kind of looking at it in kind of wider context? So like kind of the rest of the ecosystem. So like, for example, what microorganisms are potentially impacting those crops? Do you kind of consider like the whole kind of it as like a whole?
For sure. I mean, even as part of my PhD, I ended up doing some metagenomic sequencing of the bacteria that's in the root rhizosphere. People are always looking to see, I mean, yeah, I obviously, I'm focused on a lot of the genetic aspects, but I work with a lot of physiologists who, they don't particularly care what gene causes the traits. They're looking at the entire trait and how that relates to the environment. There's a lot of pushes to try and improve photosynthesis. So that's sort of like light capture, and that involves sort of the different types of light, the radiation, sort of ground cover. So definitely, I think, looking at the environment is one of a main thing to be considered, and we are always trying. I mean, agriculture has an enormous impact on the environment in a bad way. I think cows always, when you talk about agriculture, I think cows definitely get a bad rap. Everyone talks about the methane production, but the reality is that actually growing crops is a lot worse, because nitrous oxide, which is from the denitrification of all of our fertilizers that we're applying, actually pollute the airways a lot more, because I think nitrous oxide is about 300 times more potent than carbon dioxide emissions. So growing all this beautiful food for everyone to eat also damages the environment. So, reducing your meat intake is great, but if you're going to just make up for it by eating a lot more corn, you may not be doing as much good for the environment as you think you are. So like most crops, we put so much money, nitrogen and phosphorus onto the soil, but crops can typically only take up about 30 to 50% of that fertilizer, and the rest of that is either being denitrified into the air or it's being polluted into our waterways, and that can cause like algal blooms, which can really hurt lakes and waterways.
So the cows are taking the blame, essentially.
Yeah, exactly. We're just sort of hiding behind the cows. Like,
so how do you kind of go from like the research that you do in your lab, and how does that then kind of get applied to the actual kind of like farming side? That how do you kind of work with other people in the chain to kind of basically apply your knowledge?
Yeah, that's a good question. So in order to get things out, I would say a lot of the times our funding actually comes directly from, so I work mostly on cereal crops, so I'm working on grains. So a lot of our funding is from grain companies. They, or breeding companies. They want solutions, and they have their sort of areas of expertise, and we have ours. So we sort of come together, and there's a lot of linkage projects where we bring in industry projects where we do receive funding directly from industry. And we also, I mean, a lot of the people that I work with, they're always out in the field, and we work with farmers. So I'd say there's actually quite a bit of behind the scenes. And at the end of the day, I think most people in crop science definitely need to consider an applied approach, and you do want to see your research be useful. So we're always talking to farmers. What are the things they like about their varieties? What are the things they're hoping to change or improve upon? And we sort of try and find a realistic way to help them. In the sort of realm of plant science, you might get a lot sort of less applied, but definitely in the field of crop science, we have an applied end game. So we do work very closely with industry.
So the kind of genetic tools that you use, like you're kind of trying to kind of make certain traits, but how is that actually kind of done, like in terms of like the genetics, like especially like, because I always think that that's kind of like, you're obviously genetically modifying, but I think I spoke to you like recently and you said that that's not considered genetically modified, and what is considered like kind of genetically modified and what is what you're doing considered as well?
Yeah, so what is a genetically modified crop? So this is a very complicated, I'm going to try and make it as simple as possible. But it is complicated because it's also dependent on where you are and if it's going to be used for cultivation or if you're just importing it. But yeah, so the hard part is, so being genetically modified, I believe the term is just called if it's a living modified organism, and that requires two different things. It means that there's the combination of novel genetic material in the plant, which in theory would also be breeding, but the sort of the second requirement is that it has to be introduced for modern biotechnology. So a lot of the sort of new breeding technologies like Crispr and stuff are involved in that. So that basically means if I've put in a foreign gene into the plant and I've changed the genome, that's going to be GM no matter what. But with sort of CRISPR and some of these and zinc finger nucleases and talons, which are sort of called, what is their names? They're called site-directed nuclease technologies, or SDNs. There's sort of three different levels of that. So SDN1 is when you just use your nuclease to create a double-stranded break. And then the plant actually is going to repair that all by itself using its own repair mechanisms, as it would if that happened naturally. And that means that it's sort of basically the same thing as random mutagenesis. And maybe most people don't know this, but one of the main sort of breeding strategies before was you just chemically, you mutagenize, I can never say this word, but it's mutagenize. Yeah, mutagenesis, however you mutagenize the plant using chemicals or radiation. And that will create hundreds to thousands of random mutations. And you don't even really screen for it or anything. You just grow the plants out in the field and you find the phenotype that you're hoping for, and then you try and figure out what mutation caused that. So this is sort of the same thing, except I've gone in there with CRISPR and I know exactly where I've done it, and I can show you what mutation has changed. Whereas the other way breeding strategy was just completely random, and that's of course already being run out in the field. So if you can imagine, CRISPR-Cas9 technology in this sense is sort of equivalent to what's already been done, and it's really using the plant's own repair mechanism, and it would be indistinguishable from a variety that you found out in the world once you've already segregated away the input, the sort of CRISPR components that are required in order to make that transformation and the editing to occur. Whereas if you use SDN2 or 3, that's when you have to maintain. So in that one, you're not sort of allowing the plant to repair itself naturally. You're forcing the edit to happen with a template. So SDN2 is when it's a very small template, so just a little I can never say any of these words. So terrible.
Oligonucleotide.
There we go.
I'm so bad as well.
20 words, but I just read all the time. I never have to actually say out loud and sound smart. Yeah. So for SDN2, you use a small donor template, and that will help fix the guide. And that's usually like under 7 nucleotides is kind of the edit there. Whereas SDN3 is a large fragment, and oftentimes that's basically synonymous with just doing transformation and putting in a foreign gene. So you'd be just putting in a KB worth of sequence. So of course, those two are more likely to be regulated as GM.
So how are these kind of currently regulated, and like obviously kind of where you are would make more sense, but like how are they kind of like governed essentially? Yeah,
so the beautiful thing is that it's different basically everywhere. So it's going to be complicated wherever you are. But so in Australia, where I'm at right now, SDN1, which is just the natural, you make a double-stranded break using CRISPR, fixes itself. That's been deregulated. It's non-GM, which is pretty fantastic. The issue is that Australia does a lot of exporting of things, and we're not allowed to export it if the country that we want to export to does not agree with that regulation. So farmers are, of course, a bit nervous to grow these sort of crops, and I'm sure it's sort of the same way in a lot of countries. But for example, New Zealand has said no, they're GM all the time, no matter what you've done. you're using biotechnology, it's a GM outcome, sort of don't come near me. The EU has sort of said the exact same thing. If the outcome is not from natural mating or from recombination, it is GM, and that they've now actually changed that to include chemical mutagenesis. That's also now viewed as GM, even though in the past that wasn't the case. There are a few countries in Europe that are outside of the EU that are sort of okay with GM, but a lot of times they may be okay with it, but they've never actually approved a non-GM CRISPR-edited line, or they don't cultivate them at all. So the big countries who are sort of going for it would be Latin America. And I mean, that's not a country, that's an enormous set of countries, but they take up about 50% of the GM crop cultivation. So they're very pro. It is confusing in Latin America. Not all of them have said CRISPR is non-GM, but some of them have. And they're sort of trying to, at the moment, come up with a sort of a big document to really explain and have it be consistent across a lot of those countries. The US has said that CRISPR is okay, and they actually have, I think the other day was almost 150 or something GM plant varieties that have been given non-GM status, and they're growing, and you can buy them in supermarkets, such as the non-browning mushroom. So that just has a gene knocked out in it, so it has a much better shelf life. And I think everybody, would be happy about that if they, maybe not if they found out that it was CRISPR edited, but for their fridge, they're happy to have a non-browning mushroom and non-browning apples and things like that. And then in Canada and Japan, they've also said that it's non-GM. But between all these countries, even though they say it's non-GM, they all have different sort of ways of, it's all written in their legal sort of documents slightly differently. And sometimes it's a process-based thing, and other times it's a product-based way of regulating it. So some countries, I think it's like the US, they care how the CRISPR-Cas9 occurred, and that's how Australia does it. So, the template that makes it GM, whereas if there's no template, it's non-GM. Whereas countries like Canada, it's all based on the product. So they don't care how it was created, as long as there's a novel genotype or novel trait, associated with that variety, it needs to go through the Health Canada system, and then they will decide how they're going to regulate it from then on. Both have pros and cons, but I think because of all the confusion, it is hard to be a farmer, and you'd be a bit nervous if you know that you're going to export a lot of your grain.
Why do you think there is this kind of hesitancy to kind of accept kind of crops that have been that biotechnologies have been used on, and do you think that will change over time? Because obviously our population is growing, and we have to think of alternative kind of means to create kind of better food and kind of more sustainable food. But why do you think that kind of exists, and how do you think that will kind of change?
It is such a funny thing that people are so scared of biotechnology when it comes to their food, yet they're so happy to go for it when it comes to medicine. I'm sure that if people were using biotechnology and we could cure baldness, people would be taking those pills right now. There'd be no hesitation on it. But the second I tell you that I've created a mushroom that is non-browning from GM, they freak out. And I think a lot of it is that we've had a lot of really good marketing campaigns against GM foods. And that's mostly from people who are selling organic foods. I'm not trying to hate on the organic industry, but no one has ever died from eating a GM crop, but people have died from eating organic crops. So I'd like to put that out there as a fact. So safety is interesting sort of subject matter. And I think most people just don't realize that they're already eating GM foods. A lot of different industries have actually got exemptions. So the cheese industry, for example, the dairy industry, they don't have to mark anything as GM, but almost all the bacteria used to make cheeses is genetically modified. So for example, if you like cheese, you're eating it. If you've ever eaten corn or had any sort of product that has corn syrup in it, I'm going to guess that it is genetically modified. Almost, I think it's 95% of the corn grown these days is GM. And you don't have to always market that as a GM product. I think at the end of the day, I think people just don't understand what GMO really means, and I don't think they understand what DNA even is. to take it back a step further. So I can understand when someone tells you that you're eating something that has a bacterial gene in it, that might sound a bit scary, which would be an example of such as like golden rice, which is just a rice variety where people have inserted 3 different genes into it, and it means that it can now produce vitamin A. So the rice is now golden in color because it has those carotenoids in it. And that was sort of developed because people in South America and stuff were suffering from river blindness, especially children, which happens when you have a vitamin A deficiency. So they're literally trying to give sight to children, and people are resistant to eat this golden rice plant or even cultivate it, even though vitamin A is we eat it all the time, we need it, we know about it, but the plant has a bacterial gene in it. And so I think people are scared of change, and I don't think they understand that mutations are happening all the time, and that these things, and eating DNA they're in everything you're eating, and it's safe. I'm hoping that like any new technology, eventually it'll come to a point where people accept it. I mean, pasteurization in milk, for example, people did not like the idea of that. Heating up the milk, that sounded too scary. But then you realize your milk lasts 3 times as long. Like, who wouldn't want to do this? So I think once we actually get some good non-GM products out there, such as this non-browning mushroom and apples and such, hopefully we can sort of prove that it's not as scary as we once thought it was, and GM even as a whole isn't as scary as it needs to be. But yeah, I wish I understood why people were so scared, because it would help me market it as a not such a scary thing.
I mean, climate change is having such kind of a big impact like now on food production, and it's only kind of going to get worse. What role do you think that kind of crop science can play in this?
Yeah, so that's a great question, especially sort of following on from our previous question. I think so people are always hearing, right, that climate change is affecting our food production, and that's most definitely true. I think a lot of times though, it's not affecting. I mean, we can still go to the grocery store, and it's full of food. It's chocolate, it's crackers. So we're not really seeing that impact ourselves, which means that why would I try some kind of crazy new technology when I don't feel the need to sort of see it? So I think once, unfortunately, I think things sort of come about when they do. So if we start to see our food prices rising because it is harder, or if we can't get our oranges in the middle of winter because the environment is playing a role on our food, I think then maybe people will start to consider GM crops as a potential way to combat these things. I mean, we're seeing with climate change, not only just sort of longer summers, hotter summers, drier summers, but also sort of, I mean, I was in Canada recently and I was shocked at how cold it was, and we have to deal with frost tolerance, and sort of the climate's just becoming less predictable, which makes it harder for farmers to know what to grow, when to plant, and all that sort of management aspect gets really affected by it. And it's hard to sort of plan your G around the E, and then how do you manage that? I mean, I work with some really great researchers who, they have some really great prediction models, and we can try and use drones to sort of do on-farm management, but it's definitely affecting crop production, and we have to come up with solutions, and we have to do it sooner rather than later, because the countries that are suffering now it's only going to get worse.
Yeah. What do you think the kind of future will look like of crop science, and kind of what are you hoping for as well?
Yeah, I mean, as technology continues to improve, sequencing technology is already so getting cheaper by the day, it seems, and now we're sort of moving on from even understanding genes, but we're trying to look at the epigenetic control. I work with some amazing researchers who are trying to study methylation patterns in genes. So instead of just knocking out a gene using CRISPR, maybe we just methylate some of the promoter regions or remove cis regulatory elements to tweak the expression levels rather than the actual absence of a gene. And with the sort of the more big data that comes about, because now we also have so much more environmental data, so we can now really have great data sets on sort of what type of genetic varieties have been planted, what was the environment at the time, what were the yields like, and that can help us plan for next year or for 100 years from now, when you can sort of look how the environmental trends are going. We're even using satellites and phenocams and using sort of UAV, so unmanned vehicles, drones and such to help manage fields and look at weeds, and that can even help us to plan for sort of fertilizing and for chemicals that go on the plants for pest control. So before you'd go, okay, the crop's not looking so good, let's spray the whole field. But now that we can fly drones up and down, you can do a much more targeted approach, which means less chemicals, less fertilizers, better for the environment, better for everybody, much cheaper for farmers. So I guess I don't know if I know exactly what the future will hold, but I think as new technology comes about, and we're going to see a lot more engineering sort of aspects, I think, come in, and working with big data sets will be, so we need computer scientists, we need engineers, as I said, rather than maybe not just the humble biologist, but we need to sort of get even more interdisciplinary and bring in some experts from other fields.
Yeah. Thank you so much for joining me today, Karen. It's been really, really interesting. And I hope this kind of raises awareness of some of the great work that's been done in crop science as well. So thank you.
Yeah, thanks so much for having me.
Hello everyone, if you enjoyed this video, then make sure you check out some of the other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.